Hello friends, in this video we will discuss about overview of translation. Basic unit of a living organism is cell. Fundamental unit of all living organism is DNA, and DNA carry all the biological, biochemical and biophysical process throughout its life cycle. But DNA is not directly involved in carrying out biological processes inside a cell, but mostly carried out by protein. This protein is created from the information encoded within the DNA. Information contained in the strands of DNA are converted into functioning protein by two processes, transcription and translation. Now the central dogma. In order to create a new generation, the genetic information stored in the DNA has to be transferred from one generation to another. The process begins with the replication of DNA followed by transcription and translation. This involves central dogma, that is flow of information. According to central dogma, DNA makes its copy by the process of replication. DNA makes RNA by the process of transcription, and RNA makes protein by the process of translation. So, translation is the process of protein manufacturing or protein synthesis. Now the definition of translation. Process by which messenger RNA codes for a particular protein is known as translation. In translation, amino acid is polymerized to form a chain of polypeptide. Or in other words, messenger RNA is deciphered and this information is used to build an amino acid chain or polypeptide. Though DNA is transcribed into messenger RNA on the basis of their complementarity, there is no complementarity between amino acid and messenger RNA. Hence, instead of being controlled by complementarity, translation is controlled by the genetic code. Amino acid cannot read this genetic code. They are dependent on an adapter molecule called transfer RNA. Now the components of translation. There are 5 components of translation: messenger RNA, transfer RNA, ribosome, enzymes and proteins. First is messenger RNA. Messenger RNA has the description of the protein to build. It is present in both eukaryotes and prokaryotes. Transfer RNA. These are the adapter molecule. These are molecular bridges that connect messenger RNA codons to the amino acid they encode. One end of each transfer RNA has an anticodon which binds to specific messenger RNA codons, and the other end carries amino acid specified by the codons. Hence, there are specific transfer RNAs for each amino acid. Initiator transfer RNA initiates translation. There are no transfer RNAs for stop codons. This transfer RNA we have discussed in detail in separate video. You can refer my video, the link is given in the description box. Third is ribosomes. These are the manufacturing unit of cell. Translation is generally carried out inside the ribosomes. It facilitates the formation of polypeptide bond of amino acid. It serves as the platform. It provides a set of handy slots where different transfer RNA molecules carrying specific amino acid and energy molecules such as ATP and GTP find their matching codons on the messenger RNA template and deliver their amino acid. Slots are A, P and E. Transfer RNA with amino acid come together in an enclosed space, and since they are near to each other, energetically favorable conditions occur for the formation of polypeptide chains of the amino acid. Ribosomes also act as a catalyst, catalyzing the chemical reaction that links amino acid together to make a chain by the formation of peptide bond. Now the fourth is enzymes. These are required for the formation of peptide bond between amino acid and also attach amino acid to the accurate transfer RNA molecules. Fifth is proteins. They are soluble factors which are required for proper initiation, elongation and termination of translation. Now the stages or steps of translation. There are 3 steps of translation: initiation, elongation and termination. First is initiation, that is starting off or getting started. Ribosome binds to or assembles around messenger RNA at the start codon, and this start codon is AUG. Then it is recognized by initiator transfer RNA, which carries methionine amino acids. Now it is known as initiation complex, which includes the AUG codon of messenger RNA to which transfer RNA carrying methionine amino acid binds. This initiation complex is needed to start translation. Second is elongation, that is adding on to the protein chain or extending the chain. So after initiation, messenger RNA is read one codon at a time and amino acid matching each codon is added to a growing protein chain. The messenger RNA is shifted one codon over in the ribosome, exposing a new codon for reading. Transfer RNAs move through the A, P and E site of the ribosome. This process repeats many times as new codons are read and new amino acids are added to the chain. Third is termination, that is finishing up, stage in which finished polypeptide chain is released. So in the last stage, when a stop codon UAG, UAA or UGA enters the ribosome, a release factor binds to stop codon. It triggers a series of events that separate the chain from its RNA, and complete polypeptide is released from the ribosome. In this way, termination of translation occurs. Now let us understand translation with the help of this diagram. Ribosome assembles around start codon of the messenger RNA, that is the AUG codon. Initiator transfer RNA carrying the methionine amino acid comes and binds with this start codon. Thus the initiation complex is formed, which includes the AUG codon and initiator transfer RNA carrying methionine amino acid. And this initiation complex is required for the initiation of the translation. Then to this newly exposed codon, transfer RNA carrying the amino acid which matches with this codon comes and binds. The two transfer RNA are very close to each other, hence peptide bond is formed between the amino acids which is carried by these transfer RNA, and this is energetically favored. In this way, new amino acid is linked to the existing amino acid or polypeptide. Then the messenger RNA is shifted forward and new codon is exposed at the A site. Again, transfer RNA carrying amino acid matching this codon comes and binds with it, and this cycle goes on till entire codon of the messenger RNA is read. When to this A site, stop codon is exposed, then release factor comes and binds to it and the termination of the translation takes place. Newly formed polypeptide is released from the ribosome, and in this way termination of the translation takes place. After termination, polypeptide fold into the 3D shape, undergo processing as removal of amino acid, get shipped to the right place in the cell, combined with other polypeptide before it can do its job as a functional protein. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.